Good evening, everyone. And let's just wait for 5 minutes for others to join, and then we can start.
అసిక్లోఫెనాక్ ఓన్లీ అసిక్లోఫెనాక్ తీసుకో యాండో ఒక సెకండ్ ఉండు నేనే చేస్తాను కాముడు
Okay. So, good evening, everyone. I am Prashasti Pandey, PMRF Fellow and a PhD scholar from Banaras Hindu University. And I'll be taking this live session for the course Introduction to Biostatistics, whose course instructor is Professor Shamik Singh from IIT Bombay. And if you have attended previous classes in these live sessions, we will be seeing the assignment questions, previous year's assignment questions, and we'll be trying to solve them. And of course, we won't be solving this year's question, but solving them will definitely help you finish your assignments. Okay, so let's get started. Okay, so I'll just display a question and I'll give you 2 to 3 minutes. You can try solving it. If you know the answer, you can say it out loud or you can write it in the chat box, whichever is okay for you. Okay, so the first question is, in an experiment to compare gelatinase activity among three different cell lines, namely A, B, and C, using gelatin zymography, the following variances were obtained. There are three values A, B, and C for which S square value and N value is given. You have to calculate the pooled variance. So I'll give you 2 minutes. You can try solving this question. Otherwise, we'll go ahead with understanding the concept and how to solve these kind of questions. if you can solve this question, you can write your answers in the chat box. Also, this kind of question is a numerical question where you have to write the entire answer in decimals in this question, and it's not a MCQ type of question. Okay, so let's try solving this. So first, let's try to understand the concept because the question asked about the pool variance. So let's try to understand what pool variance is. Okay, so pool variance is a method which is used to estimate the common variances of two or more groups when it is assumed that all groups have same variances. Okay, so all groups having same variance is called as homogeneity of variance, right? So pool variance is nothing but a method which can estimate the common when you have multiple groups, okay, when you have more than one group and there is all the groups have same variances, then in that case you can calculate or you can estimate the common variances of all those groups, and that common variance is termed as our pooled variance. Okay, so basically it combines the variances from multiple samples into a single weighted and average variance. Okay, because so when you have multiple groups, say group A, B, and C, you can have variance of each of this, right? And then you combine all this and then create one variance. So this one variance value will have uh, the average values of all A, B, as well as of C. Okay, so that is how it combines the variances of multiple samples. And pool variance is commonly used in your t-test, in ANOVA, and in various other statistical analysis when we are doing a comparison between different groups. Now, when to use pool variance? So when we are comparing two or more groups and we are assuming that the population variances are equal. Okay. Then second, when we are you are doing an independent t-test or we are doing ANOVA, in that case also pool variance is used. And third, pool variance can help us better estimate the variance by combining it from multiple groups. Okay. So now coming back to our question, the data that was provided was for cell line A, B, and C. We have the variance 6, 6, and 8, and the sample size was 5, 7, and 8. Okay. This was our value. Now the formula for pooled variance will be this, right? So now you to solve this kind of question, you have to calculate the individual values. Okay. So now for here, like for this one, you need to calculate n minus 1 into S1. So this is how you calculate the individual value. You just take this small part of the formula. Okay. This will be same for all the three. So n2 minus 1 and variance of B cell line, similarly N3 minus 1, and variance of the third cell line. Okay, and then you sum up everything and then divide it by N1 minus 1, N2 minus 2, and N3 minus 3 summation. Once you calculate the individual values, you just need to put everything in this. So when we calculate N1 minus 1, so N1 is 5. Okay, so that's what 5 minus 1 into variance is 6, and then you get the answer 24. Similarly, for B cell line, your N is 7 and your variance is 6. So 7 minus 1 into 6 will be 36. And for your C cell line, N value is 9 and variance is 8. So then your value comes to 64. Now, once you have all the values for the individual parts of the entire pooled variance formula, then you just need to put the values. Okay, so our values were 24 plus 36 plus 64, which comes to 124, and you just need to now you just need to add, this was the summation of the nominator. Now you just do the summation of the denominator, which was 4 plus 6 plus 8, which is 18. This is denominator. Now you just need to calculate the pool variance, which is your 120. Oh, I'm sorry, this is 124. So 124 by 18, which is approximately 6.89. Okay. So the pool variance for the three cell lines, your answer is 6.89. Okay. Since it was a numerical question and not an MCQ question, for these kinds of questions, you have to type the answer in the answer box that is given in this question. Okay. So I hope this was clear. Now let's move on to our next question. In a study, subjects are randomly assigned to one of three groups: control, experiment A, and experiment B. After treatment, the number of subjects showing improvement and the numbers of subjects not showing improvement in each group are recorded. The appropriate statistical test for comparing counts is: options are t-test, correlation coefficient, chi-square, and analysis of variance. You have 2 minutes, you can solve this question. It's a theoretical question, should be easy. Alright, so let's try to solve this question. We will understand the concepts first and then come up to the answer. Okay. So there were 4 tests in the options, and we will understand each test one by one so that we can understand the answer. So first was t-test. So t-test is a statistical test which is used to compare the means of two groups. Okay, and determine if the observed differences are statistically significant or no. So if we have two different groups and we have the means of them, then Then to say whether the mean value or the average value of these two groups are whether they are statistically significant or not, to test that in statistics can be done by using a t-test. Okay. So this test is widely used when the sample size is small and the population standard deviation is unknown. So to calculate t-test value, you don't need a standard deviation value and the sample size should be small. In that case, you can compare the mean values of the two groups that we are trying to compare and come up to a conclusion whether these two groups are significantly different or not. There are various types of t-test. It can be independent sample t-test, paired sample t-test, or one sample t-test. In case of independent sample t-test, it will compare the means of two groups which are independent. And for paired sample t-test, it will compare the means from the same group at different time points or under different conditions. So the group of objects in that in the comparison will be same, but it could be at different time points. For example, if you're trying to treat the plants with stress, you will have the same 10 plants, but then you will compare the mean of suppose radicals formed in the plants at 0th hour, at 3 hour, at 9 hour, at 11 hour. So the groups or the means will be compared from the same group but maybe at different time point or at different condition. So that is called as paired sample t-test. And one sample t-test will be comparing the mean to an to a known or a hypothesized population mean. So in this case, when we are comparing the two means, the one of the groups will have a hypothesized population mean and then the other one is a calculated one, and comparing these two means. So in this case, we are trying to say that whether our calculated mean value is statistically same or different from the hypothesized mean value. That's when one sample t-tests are used. Next is our correlation coefficient. So correlation coefficient basically measures the strength and the direction of the linear relationship between two variables. There are different types of correlation coefficients can be used, but the most common one is Pearson's correlation, which is denoted by R, and the value of the R can be either equal to 1, R can be more than 1, or R can be less than 1. In case of more than 1, it will be a positive value, and in case of less than 1, it will be a negative value. So when the R value is equals to 1, the relationship is a perfect positive relationship. When the R value is more than 1 or it is in positive direction, the correlation is positive. If it is less than 1, it is a negative correlation. If it's 0, then in that case, there will be no correlation. Okay, that means the values that we are trying to correlate are not correlated to each other. But when there is a positive correlation, in that case, increase in one value will cause increase in another value. And in case of negative correlation, increase in one value will cause decrease in the other value. So these are called as correlations, and R is the correlation coefficient. Now, the third one was chi-square test. Okay, so chi-square test is a statistical method that is used to determine if there is a significant association between categorical variables or whether the observed frequencies differ from expected frequencies. So now to calculate, so now if our data is in form of category, okay, in that case, we cannot use t-test or we cannot use these kind of statistical tests uh, where categorical values are not considered. So in that case, to compare categorical values and whether the frequencies that we have observed is different or is same from the expected frequencies, we use chi-square test. Now there can be two types where the chi-square test is used. One is test for independence, and the other one is goodness of fit test. So chi-square test of independence will test if the two variables are independent or they are related. On the other hand, goodness of fit test will observe whether the distribution that we are showing fits the expected distribution of that particular data set. Okay, so this is what chi-square value tells us. Now this key points with chi-square value are that they are used with categorical data. It will compare the observed frequencies to the expected frequencies, and to calculate the chi-square values, relatively large sample size is always required. Unlike for t-tests where small sample values also work. And also the chi-square value will always be a non-negative value because it is based on the squared differences. The values of your expected and observed are always squared, and the differences between them are uh, the difference is squared actually, so you will never get a negative value. The values will always be positive. There are certain assumptions with using chi-square, that the data should be from independent observations, categories should be mutually exclusive, and expected frequencies in each cell should generally be at least 5 or for valid approximation. So to summarize, chi-square value will help us analyze the relationships in categorical data. It is widely used in scientific research, surveys, and genetics and quality control. It will test whether the observed data fits the expected data, and will check the degree of freedom and assumptions before interpreting the results. Now, the fourth option was ANOVA, which is also called as analysis of variance. Now, ANOVA is also a statistical test, but it will compare the mean value of 3 or more groups, and it will see whether at least one of the groups of the ones that we are comparing either significantly differ from each other or not. Okay, so unlike in all the tests, so in case of t-test we can only compare two values. For chi-square test the data should be categorical, but for ANOVA there can be 3 or more groups. You cannot work with 2 groups in case of ANOVA. There has to be more than 2 groups, like 3 or more groups. And when we compare the means of 3 or more groups, if we need to see whether any one of those groups are significantly different from the others, then in that case we use ANOVA. Now, why ANOVA? Because this is exactly what I told, that when 2 groups are used, then t-test is sufficient to test the 2 groups differences. But when there are more groups, you cannot use multiple So basically ANOVA is like comparing multiple t-tests. So if you have say 3 groups and you are comparing the mean of A and B and showing whether they are significant, then you are comparing B and C and showing whether they are significant, and then you are comparing A and C and showing whether they are significant. So instead of doing multiple t-tests, what you can do is you can simply use ANOVA, and it will compare all these 3 groups at one time and tell us whether any of these groups are significantly different from each other. So it becomes much more easier, and using multiple t-tests will also increase the chance of type 1 error or chances of false positives. So basically ANOVA will help test all the groups simultaneously. As I told you, you don't need to do multiple t-tests, and it will have more control over the errors. Now there are few assumptions for using ANOVA. First is that observations of all the values that are being used should be independent. The data should be normally distributed and the variance across groups should be similar. When these conditions are fulfilled, ANOVA is the right choice to test the differences between 3 or more groups. So, coming back to the question, subjects are randomly assigned to one of 3 groups. This should be the cue that they are talking about 3 groups: control, experiment A, and experiment B. So in case of using 3 groups, and if we have to complete that, we have to analyze that after treatment, the number of subjects showing improvement and the number of subjects not showing improvement in each group. So basically, when you are taking 3 groups and you have to compare the 3 values, whether any one of them is being different from the other two, the best statistical test should be obviously analysis of variance or ANOVA. Okay, so I hope the concept to this question was clear. Maybe we should focus more on understanding the concept behind the questions, and then we can easily solve the answers. So let's move on to our next question. In an experiment to compare protein expression levels among 3 different treatments, control, drug A, and drug B, using Western blot, following sample variances were observed. And you have the values for the sample variances and the n value. And yes, we need to calculate the covariance, which is not given in the question. I will just write it here. Okay, and we need to find out what is pool variance. I'll give you 2 minutes. This question is very similar to the first one that we have already solved, so it should be easy. Let's just write down. So we by now know what a pooled variance is and what the formula for pooled variance is. So we have three groups: control A, drug A, and drug B, and we have variances of all these and the sample size of all this. So to solve this, we just need to use the same formula, which was this. So we just need to use the same formula, and we will just compute the Ni minus 1 and our variance time. Okay, so same here in this case for our control group, our sample size is n, so it will be 5 minus 1 into the variance is 4. For drug A, it will be 5 minus 1 into 6, and for drug B, it will be 5 minus 1 into 8, and all these values will be calculated easily: 16, 24, and 32. Okay, now we just need to add everything. So in same way, when we add for numerator, we just need to add 16 plus 24 plus 32, which will be equal to 72. For denominator, we just need to add all the values of n minus 1, n 1 minus 1, n2 minus 1, and n3 minus 1, which is same here. So it should be 4 plus 4 plus 4, which is 12. Now we just have to divide them. So it will be 72 upon 12, which is equal to 6. Okay. So the pooled variance for this will be 6. Okay. So I hope it was easy. So let's now move on to our next question. Okay, so answer to this question is 6, which is option A. Okay. So in an experiment to compare the protein expression levels among three different treatments, control, drug A, and drug B using Western blot, the, the appropriate statistical test to determine if there is any significant differences in protein expression levels among these groups is. Your options are ANOVA, t-test, correlation coefficient, and chi-square. We have just discussed this question. If anyone knows the answer, you can answer it. So let's solve this, and this is easy. And this previous question, because we have to calculate the differences between three treatments, that is control drug A and drug B. The answer will be analysis of variables. Okay. Let's move on to our next question. let's solve this. So TSS is basically total sum of square. So it is another statistical measure that can quantify the total variation in the observed data. Okay, so basically it will show that how much an individual observation will deviate from the overall mean from the data. So suppose if this is the mean value and your x and y are the two observations from the entire data whose mean value is this, and your x value is lying here from the mean and your y value is lying here from the mean. Okay, so basically your TSS will quantify the difference of how far your x is lying from the mean and how far your y is lying from the mean. Okay, so it will show that how much the individual observations will deviate from the overall mean of the data. Okay, so now the definition for total sum of squares is that it is calculated as the sum of squared differences between each observation and the overall mean. Okay, so this is the formula for total sum of squares, where it is summation of the square of differences of individual values from their mean values. Okay, where your xij is the observed value for treatment. So for in case of this is an example that in case of treatment of j in block I, then your x will be the observed value, your x bar is the overall mean, and the summation runs from all the observations. Okay, so now when it is telling you that how much variation is there in the data as the whole, so in experimental design, sum of squares can be decomposed into formats. One can be sum of squares due to treatments and the other can be sum of squares due to blocks. Third one is error sum of square. Now, sum of squares due to treatment is that how much variation will be there or how much variation can be explained by the differences between two different treatments. Whereas sum of squares due to blocks is that how much variation will be there when two different blocks are considered. And error sum of squares will be how much variation is unexplained or random error. So your total sum of squares will be a sum of treatments plus sum of squares of block and sum of squares of error. When you combine all the sum of squares, then you get the value of total sum of squares, which is your TSS, which was the question. So now, if in case of analysis of variance or regression, your total sum of square will be your sum of some variation explained by your regression, which is SSR, which will be either due to treatments or predictors, and SSE, which is the unexplained error or the error of the residuals. Okay, so total variation will be the sum of variation which will be explained by this model, and this model, which is SSR, in case of either if you are using regression, then SSR will be the variable. In case of ANOVA, it will be something else, but your total sum will be the variation because of that model that you are using and the unexplained variation, which is error or residuals which comes in the data. So when you sum that up, you will get the value of TSS. So according to this question, your TSS will be SSR plus SST, which is your option 2. Okay. So I hope this was clear. So the next question is, what is the estimate of common variance MST? session. Let's first understand that what question was exactly asking. It was asking about MST, which is also called as mean square error. Okay, so mean square error is an estimate of common variance, that is the variance within the groups or the error variance of the data, and it represents how much variability is left unexplained after accounting for treatments or blocks. Okay, so according to definition, the formula for mean square error will be your sum of square errors divided by degree of freedom of errors. So as we have learned previously that the sum of square of errors is the unexplained errors. Okay. The other one was the error which was created due to the treatments or due to the condition of uh, the model that you are applying. So that will be the other error that is called as SSR, and SST is the sum of squares of the error which is unexplained. So when you divide this SST by the degree of freedom of error, then what you get is MST, which is called as the mean square error, and it is the estimate of the common variance which has not yet been explained or whose variability is unexplained after accounting for treatments. So MSE is the error of variance per degree freedom, and it is used to assess the consistency of data within the groups. Okay. So now why MSE is important? So MSE will provide an estimate of the variability that cannot be explained by the treatments or the block. So when any experiment is performed in suppose three different groups, and group A was control, group B was on treatment, and group C was on treatment, then because of treatment of group B and C, there there was some variability in the control as well where there was no treatment. Okay, so the variability coming in the control group without any treatment or without any block which is not explained. So to estimate that variability, MSE can be used. Second, it is also used to calculate F statistic in ANOVA test, which will help us compare different treatments of the effect. And third, the smaller the MSE value is, the that model will be able to explain most of the variability. Okay. So now, from whatever we have understood by now, MSE is an estimate of common variance, and it is calculated by dividing the sum of square of errors, which is SSE, by corresponding degree of freedom. Okay. Now, so SSE is the unexplained variance, N is the total number of observations, and if K is the number of groups or treatments, then your degree of freedom will be N minus K. Okay, so that means that MSE can also be written as SSE divided by N minus K, where N is the total number of observations and K is the number of groups or the treatments, which is the formula for degree of freedom. And this is the average squared error which per degree of freedom, and it is widely used in ANOVA or in regression analysis. So now the answer of our question will be NSE divided by N minus K, which is your option 1. Okay, so that is the formula for MSE. Okay. So our next question is, if there are B number of blocks and K number of treatments in the population, then how many samples need to be? Okay, so the question is asking that if there are B number of blocks and K number of samples, how much samples there should be to get us to this number? Okay, so let's try solving this question. So the question is talking about block designs. Okay, so in case of block designs, each block will contain all the treatments. So that is how a block design is done. So in that case, the total number of samples or the experimental units will be number of blocks multiplied by the number of treatments. So here in our question, number of blocks was B and number of treatments was K. So total number of samples will be B into K. So if you can take it for an example, that if there are 3 blocks and there are 4 treatments in each block. Okay. So the total number of samples will be 4 into 3, which is equal to 12. So that is what our question was asking, that if there are B number of blocks and K number of treatments in the population, then how many samples should be there? It should be B into K. Your option C is correct. Okay. So our next question is, in a study examining the relationship between the concentration of neuraminidase and the response of diabetic wound healing, the regression equation for was derived from the data y equals to 980 minus 0.56x to see how x and y are related. Your options are independent and not related, negative correlation, direct, and exponential. So, in case of regression analysis, it is basically a statistical technique that will help us understand the relationship between one dependent variable, which is the outcome, and one independent variable, which is the predictor or the explanatory variable. Okay, so regression analysis will basically help us predict, explain, or model how the changes in the invariant independent variable will influence the changes in a dependent variable. So, according to this, the key goals of regression analysis will be prediction, which is to estimate the value of the dependent variable based on the values of unknown independent variable, and explanation, which is to understand how changes in predictors can affect the outcome, and modeling relationships, which will quantify the strength and the direction of the associations. So there can be different types of regressions. It can be simple linear regression, multiple linear regression, or non-linear regression. For simple linear regression, there has to be one independent variable and one dependent variable, and the equation will be y equals to a plus bx, where y will be a dependent variable, x will be an independent variable, a will be the intercept, and b will be the slope or the coefficient. For calculating multiple regressions, there has to be more than one independent variable, and it will be calculated by y equals to a plus b1 x1 plus b2 x2 plus however much your variables are. In case of non-linear regression, we will calculate the relationship which is non-linear. For example, it can be either exponential or it can be logarithmic. So now in our question, the equation was y equals to 980 minus 0.565x. So here y is an independent variable. 980. Okay, so y is a dependent variable. 980 value is the intercept, and 0.565 is the slope, and your x is the dependent variable. So when x equals to 0, y equals to 980, and the slope is minus 0.565. That means for every 1 unit of increase in x, y will decrease by 0.565 units. That means because one is increasing and the other one is decreasing, this is a negative relationship. So, okay. So that means the answer to this question is where y is equals to 980 minus 0.56x, x and y are related in a way of a negative correlation. Okay. So next question is, which of the following experimental scenarios would benefit from using a randomized block design? Your options are A, population is large, B, population is highly heterogeneous, C, population is reasonably homogeneous, and D, population can be grouped into subpopulations. Okay, so to answer this question, let's first understand what randomized block design is. So RBD is an experimental design that is used to control for known sources of variation among experimental units by grouping them into homogeneous blocks before applying the treatment. So it is especially useful when factors other than the treatment might influence the outcome. And the features of RBD will be blocking, where experimental units are divided into blocks which can be based on any factor that is expected to influence the responsive variable, for example, age, soil type, or location. Then the other feature is the randomization, that within each block, treatments are randomly assigned to ensure unbiased results. Then there is homogeneity within the blocks, that even though the treatments inside a block are randomly designed, the units within the block are similar. That will reduce the variability within blocks and improving the accuracy of treatment comparison. And fourth one is comparison within blocks. The differences among treatments are compared within the blocks, helping control for block effects. Now, let's take an example to understand randomized block design. That when if we are investigating how different fertilizers can affect plant growth, and the problem is that soil types will vary across different fields, then in that case, the solution will be to group the fields into blocks based on soil types. And within each block, randomly fertilizers will be assigned to the plots and then analyze the effect of the fertilizers while controlling for differences due to soil type. So, in that case, a randomized block design will be used when you can group the experimental units into block based on similar characteristics that may affect the outcome, and this blocking will help control the availability caused by known factors, and each block is relatively homogeneous internally, but different blocks may vary. So the design is most useful when you can group the population into subpopulations that are expected to differ. Now, coming back to the question, that which of the following experimental scenarios would benefit from using randomized block design, the answer will be population can be grouped into subpopulations. Okay, that was the last point in understanding the randomized block design. So our last question is, in an analysis of variance ANOVA, comparing the motility of E. coli across four different treatment groups with a total sample size, computed F statistic is 2.33. In this scenario, what can we infer about the p-value? So this question is about ANOVA, the F statistic, and the p-value. So now this is a numerical type of question, and let's understand this. So the explanation will be that F statistic value is given in the question, which is 2.33, and it is used to determine whether there will be any difference between the group means and whether that difference is statistically significant or not. So now whether the mean between the two groups are statistically significant or not will depend on whatever the F value is given, the degree of freedom, and the P value. Now how do we calculate degree of freedom? We have learned previous that degree of freedom will be calculated by two values, which is number of groups and total sample size, which is n, and the number of groups. So in this case, the number of groups is 4, and n minus 1 here is 4 minus 1, which is equal to 3. So number of groups uh, here is 3. Okay, and total sample size. So total sample size is 78. Okay, and to calculate the total sample, to calculate the degree of freedom of total sample size, it will be 78 minus 4, which is equals to 74. So then the degree of freedom will be 3 comma 74. So now moving forward, to interpret the F statistic for this, for this, that means the degree of freedom that we have calculated, which was 3 comma 74, the critical value of F at 0.05 significance, which means 95% of confidence. So at 0.05, significance level is approximately 2.73, which is given in the question. Sorry, 2.73, which is given from the standard table for this. And because the computed value is 2.33, and it is less than 2.73, right? It is less than the critical value, which is 2.73. So that means that our p value is greater than 0.05 and we fail to reject the null hypothesis. So in our question, whatever the null hypothesis was, we will reject that null hypothesis, and that null hypothesis would have been that there was no difference in the motility of E. coli across the 4 treatment groups. So the answer will be to the question was asked about to understand the inference of the p value, and because of the f statistic being less than the critical value, which is 2.73, the answer will be less than 0.05, which is your option C. So I hope these questions were clear for today, and this was the last live session for this course, and I hope you were able to understand all the questions. If there are any doubts, you can ask, or otherwise we'll end the session, and all the very best for your exams.